In the previous movie, we saw how synapses can be strengthened by high rates of synaptic activity, as was demonstrated by the phenomenon of long-term potentiation. In this movie, we show the reverse of this process, whereby synapses can be weakened by a phenomenon called long-term depression. In contrast to long-term potentiation, long-term depression is the weakening of one synapse due to low rates of activity at that synapse. Long-term depression is demonstrated in the same way that we demonstrated long-term potentiation, that is, by recording postsynaptic activity after stimulating one of the inputs to the postsynaptic cell. We begin by showing the control response evoked by a single electrical stimulus delivered to the presynaptic neuron. There's the control response. Next, the presynaptic neuron is repeatedly stimulated, but at a low rate of stimulation, at about one stimulus per second for several minutes. We stop for a few seconds and then stimulate the presynaptic cell again with a single electrical pulse. Now the response evoked in the postsynaptic cell is substantially smaller than the control response, the response that was evoked before the low rate of stimulation was applied. The smaller evoked response is an expression of the synapse that has been weakened. Now let's look at the depression as it emerges over time. And the first thing we're going to do is stimulate with a single electrical pulse. And that's going to evoke the control response. And we're going to stimulate at very long intervals, maybe 30 or 40 seconds between each pulse. And that's going to give us a series of control responses. In fact, we're going to do that for about 10 or 15 minutes. And the control responses are shown as the black dots. Now let's look at a blown-up version of the synapse. The presynaptic cell will be driven at a low rate of about 1 pulse per second for about 10 or 15 minutes. But I want you to pay attention to the enlarged version of the synapse. And the reason is that each action potential that arrives at the synapse will open both AMPA and NMDA receptors. The receptors are not shown in the figure. But the key feature is that the opening of the NMDA receptors will evoke a small calcium influx into the postsynaptic cell. The rate of stimulation is so low that calcium is cleared from the postsynaptic cell and is completely removed before the next action potential arrives, which then evokes another calcium influx. In other words, there is no buildup or lingering of calcium between synaptic events. The importance of the small calcium signal evoked by each action potential, which is short-lived in the postsynaptic cell, will become apparent in the next movie that describes the structural and functional features of CAM kinase 2. But here is an abbreviation of those events. After stimulating at a low rate for 10 or 15 minutes, we're going to stop for a moment and then present a single electrical stimulation again. The response is much smaller because the synapse has been weakened. To show that the synapse remains weakened for more than an hour, this presynaptic cell is again stimulated, but now with a 30 or 40 second interval between each stimulus, and the responses are recorded and are shown as the blue circles in the lower graph. So long-term depression is demonstrated by the difference in the amplitudes of the control response, shown as black circles, and the responses evoked after the low-frequency stimulation, as shown by the blue circles. The difference is indicated by the arrow labeled LTD. In this slide, we demonstrate the events that generate synaptic weakening and long-term depression. First, there is a small influx of calcium that activates a small amount of calmodulin. The relatively small amount of calcium-activated calmodulin then activates phosphatases, enzymes that remove phosphate groups from proteins. One phosphatase that plays an important role in synaptic weakening is called calcineurin. The activated phosphatase then does two things. First, it causes AMPA receptors to be removed or sequestered from the synapse. And second, it dephosphorylates the AMPA receptors and thereby reduces their conductance. Remember that reducing the conductance means that less sodium ions pass through the channel when the channel is open. Stated differently, synaptic weakening has the exact opposite effects of synaptic strengthening. Indeed, synaptic weakening due to prolonged low-frequency stimulation can actually reverse the synaptic strengthening that was produced by a previous period of high-frequency stimulation. These processes, then, are essentially the yin and the yang of synaptic plasticity. In the next movie, we will see how and why the structural features of CAM kinase 2 enable large calcium signals to affect the kinase in an entirely different way from the effects that small calcium signals have on the kinase.